It's really an honor to be here in front of this group and share my research. I'm an assistant professor in the Department of Molecular and Cell Biology at UC Berkeley, where I've run a lab and taught undergraduates and graduate students for about 5 years. And today I'm going to tell you a story about tuning mitochondrial function to cellular needs on the micron scale. And here on my title slide, I've included the Berkeley motto, "Let there be light." And this is a motto that we take as inspiration in my lab because we're very focused on microscopy as a tool to study cells and metabolism, but also because we are big proponents of open data and sharing our data with the community, letting light shine on the achievements of Black and brown students and to share our discoveries with the world. So on this title slide, there's an image of a cell. And for those of you who are not cell biologists, you might be looking at this image and not understanding exactly what's going on here. And it might just look like something bright and colorful and doesn't have a lot of meaning to it. This is a human fibroblast, and this blue circle represents the nucleus of the cell. And all of these green filaments are actually the mitochondria. So those of you who have had a bio class or maybe took AP Bio, what do you know about mitochondria? The powerhouse of the cell, right? That's the stereotype that most people remember, but mitochondria are actually much more complicated than this, and I'll tell you a little bit about that today. So going back to that AP Bio class, this is probably the more typical view that you could remember of what a cell looks like. Here's our nucleus where most of the genetic material is, and then there are all these different compartments that are the different organelles. And I'm really interested in the mitochondria, and they're unique because they have their own genome that is separate from the rest of the DNA inside of the cell. Mitochondria are really important because damaged mitochondria or degeneration in mitochondrial function leads to chronic disease. And we know that mitochondrial quality declines with age, and this contributes to many different chronic and genetic diseases in humans. Mitochondria are the most critical in tissues like muscle and in neurons that have a really high energy demand. So these cells need a lot of chemical energy in the form of ATP to perform their functions. And when there's a damage to mitochondria with age, this leads to a decline in function. And this is an additive damage because not only will there be a decline in the muscle, there will also be a decline in the neurons that innervate the muscle, and this contributes to chronic symptoms of aging like loss of gait and stability, but also to genetic diseases like Parkinson's disease and motor neuron diseases. So mitochondria are also really implicated in genetic disease, and defects in any of the genes that are required to build mitochondria leads to highly heritable degenerative diseases that recapitulate the symptoms of aging. So what does that mean? Here I'm showing you 2 mice. On the top is a wild-type mouse, and on the bottom is a mouse that's the same age as the mouse on the top, but it has a mutation in one gene that is essential for mitochondrial function. And so this mouse has hair loss, loss of movement, it's a fatter mouse, it has sleep problems, it has many of the problems that accumulate with aging. So what this means is that we can recapitulate some of the key features of the mitochondrial dysfunction using genetic tools by understanding what are the different genes that are required to build and maintain this organelle. So this is the classic powerhouse view of the mitochondrion. You have your favorite carbon source going in, and then you have energy in the form of ATP coming out along with byproducts that are carbon dioxide and water. So when we breathe out CO2, that CO2 is actually coming from all of the mitochondria in your body, and that is how we're getting rid of that waste product. However, if we move beyond that classic powerhouse model, we now appreciate that there are many different biochemical pathways that require mitochondria that are not only linked to the production of ATP. So this figure is really complicated, but what I want you to take away from it is that there are many different pathways that are moving in and out of the different parts of the mitochondria, and these are important for making nucleotides, sugars, amino acids, and lipids that are required for cellular growth. So what this means is that cells require mitochondria not only for the energy production and the bioenergetic homeostasis, is how we say that, but also for producing these building blocks for cell growth and division. And because of that, even cells that don't need a lot of ATP from oxidative phosphorylation require mitochondria. One quite famous example of this is in the context of kidney cancer. So these cancer cells are highly glycolytic, which means they mostly burn sugar. They don't really rely on their mitochondria to produce energy. However, they do rely on their mitochondria to make these other molecules that they need for metastasis. And so if we look at patients that have a lower mitochondrial function, they actually survive longer than the patients that have the higher mitochondrial function, because in this kidney cancer, those high functioning cells are able to move to other locations in the body and establish more tumors. So one of the ways that we visualize mitochondria is using a technique called cryo electron tomography, and I believe there was a talk that touched on this yesterday. But we use specialized microscopes to shoot electrons at a sample that contains cells and mitochondria, and then based on the density of the images that we get, we can make reconstructions of what the proteins and the membranes in the cell look like. So in a mitochondrion, there are multiple membranes. There's the outer membrane, which is labeled OM, an inner membrane that forms these tubular cristae structures. And this ultrastructure is really important for the localization of the proteins that make ATP and make all those other molecules that I mentioned. So if we take this image and we actually look at the back projection of where these different proteins are that we care about, we can see that actually the inside of the mitochondria is really crowded with these different machines, and we call them mechanico-chemical machines because they burn energy, they generate energy, and they physically move using rotors like tiny machines that are embedded into the membrane. So here, this molecule ATP synthase is a key motor that pumps protons that is important for establishing a gradient for making ATP in mitochondria. So that's blue. And we can see that there are many, many of these motor proteins decorating the cristae, right? So inside of this environment, it's crowded, but it's also highly structured. And that links the functions of the proteins and their localization to the membranes. Mitochondria, as I mentioned, are special because they have their own genome, and that's because they're the product of something called endosymbiosis. So many billions of years ago, there were cells called archaea that didn't have mitochondria, but they did have lots of proteins that are important for energy production and for maintaining their internal structure. Over time, these cells engulfed bacterial cells, like prokaryotic cells, that became the endosymbiotic organelles like mitochondria and the chloroplasts that plants use to generate energy from sunlight. And it was really this acquisition of these organelles, this endosymbiosis, that powered the increase in cellular complexity that was needed for life to go from being single-celled to being multicellular. And the result of that is that because this proteobacterial ancestor became the mitochondria in our cells, they had their own DNA, and our mitochondria have their own DNA. Now we know, based on different genetic analyses, that the bacteria that became mitochondria had many, many genes in their genome. But today, our mitochondria contain only a few genes. Most of the genes were transferred to the nuclear genome, and then there were a few other proteins that are now used to help maintain mitochondria that came from other sources. So I wanted to just ask a question, based on what you know about mitochondria, which might only be what I've told you, what proportion of the total proteins inside mitochondria do you think comes from the genome inside versus the nuclear genome? Does anybody have a guess? 20%? Okay, other guesses, more or less? 100%? What else? 1%? So the answer is 1%, okay? So in human cells, we know that there are about 1,100 different proteins that are needed to build functioning mitochondria, and there have been 3 major studies of the mitochondrial proteome that all converged on a pretty similar number. But in human cells, there are only 13 proteins and some different functional RNAs, like transfer RNAs, that are needed for their expression. And so this means only 13 out of 1,100, or about 1%. So why do you think cells would expend energy maintaining a separate genome that only encodes so few proteins? Well, the rationale is that these proteins are so critical and they need to be locally regulated close to where energy production and biosynthesis are happening, and they can't be transferred to the nucleus. And this idea is supported by the fact that there are many different mitochondrial genomes in each cell. So there are many backup copies. It's not sufficient to have one copy or to have two copies the way that we have a diploid nuclear genome. There are hundreds to thousands of copies of mitochondrial DNA in each one of our cells. Now, I'm gonna skip over some of this because previous work from my lab has shown that there are dedicated proteins that regulate the mitochondrial genomes and move them between two different states, a quiescent state where they're not really doing much and they're tightly packaged up with proteins, or an open, active state where different transcription factors and replication factors can engage with the DNA and express those genes, leading to the production of new electron transport chain complexes. But that is a static view. The other thing that we're known for is live fluorescence microscopy. So here I'm showing you a human cell. This is an osteosarcoma cell, or bone cancer cell. And what we've done is we've introduced a blue fluorescent protein. So this is a protein that normally is found in jellyfish, and it fluoresces blue. And we took that gene and we introduced it into the matrix of the mitochondria, which is the interior compartment. And so now, using our microscope, everywhere that we see these blue filaments, we can see where the mitochondria are and what they're doing in the living cell. And we found that there are many proteins that regulate the movement and motility of the mitochondria in the cells. And one of the most important is called DRP1. Here, I tagged it with this red fluorescent protein that we call cherry. And so we can see everywhere that this protein is interacting with these tubules of mitochondria in the live cell. And it turns out that this protein is really important for the mitochondrial division process. So these mitochondria are constantly fusing with each other, dividing from each other, and sharing their contents, including the DNA, the RNA, and the proteins. So this protein is called a dynamin-related GTPase. And it forms these helical structures that we can see by electron microscopy. And we can use molecular dynamic modeling to see how we think that these form helices around individual tubules of mitochondria and hydrolyze GTP to pinch them and divide them. A big open question in the field when I got into this area of research was what determines where this actually happens. So are these mitochondria dividing randomly, or are there signals that regulate that process? We knew, again from fluorescence microscopy, that this protein didn't just bind to the mitochondria anywhere. So here, the outer membrane I've labeled with a red fluorescent protein, and our protein DRP1 is labeled with a green fluorescent protein. And we can use super high resolution microscopy to look at the binding of individual molecules to the mitochondrial tubules and see the formation of the early steps of those helices that I mentioned. And so what we found is that they're binding not just anywhere, and they're also hanging out on the mitochondria for a certain amount of time. So after division has occurred, we can see this mark, which is almost like a scar that tells us that recently the mitochondria has divided. So my claim to fame, I guess, is that in my own work, I made the link between the regulation of the mitochondrial DNA on the inside and this protein DRP1 on the outside. So these are frames of a movie. And on the top, there are three colors. On the bottom, we're looking only at the mitochondrial tubule. Here it's blue, just so that you can see when it's dividing. And so here's our mitochondria. Green is showing us a mitochondrial DNA binding protein, so we can see where the DNA is on the inside. Red is showing us DRP1, so we can see where it's forming a helix on the outside. And what I found is that this protein DRP1 is recruited to the places where the genome is replicating on the inside. And this was extremely important. If we measure the intensity of fluorescence for these different reporters across this tubule, what we find is that there's this peak of fluorescence that tells us where that DNA protein is. And then we see that there's peak of fluorescence for DRP1 arrives after, and then there's division of the mitochondria. So this told us that there are signals that are coming from the mitochondrial genome replication process on the inside that are recruiting the division machinery on the outside. And this was really radical because up until this point, everyone assumed that these proteins like DRP1 in the cytoplasm were controlling the mitochondria. But this showed that there are signals that go the other way, and it's more of a two-way communication between the organelle and the rest of the cell. And so this led to the idea that mitochondria can integrate signals that are coming from other compartments or the cytoplasm that could influence the expression of the genome, and then there's an output that can be read as the morphology of the network. So similar to organ networks or cell networks that allow higher cognition in the brain, we predict that there are these biological networks that take inputs, process them, and lead to specific outputs for mitochondria. And that this can be visualized by the shapes and the behaviors of the individual organelles that will tell us about the cellular states and fates. So not only what is the metabolic of the state right now, but based on how the mitochondria are moving, can we make a prediction about what is going to happen in the future? So in my lab, one of the things that we're really focused on now is molecular phenotyping, where we're systematically profiling different inputs into mitochondria and then measuring the outputs. And we use fluorescence microscopy to do this using a technique called metabolic labeling, where we feed the cells fluorescent fatty acids or nucleosides or amino acids, and then we ask when there are changes to the inputs, how do these types of outputs change? And that tells us about mitochondrial function. One example is that we can look at mitochondrial gene transcription and translation by pulsing in a synthetic amino acid, which is recognized by the transfer RNAs inside mitochondria, and then this leads to the production of nascent peptides that incorporate this marker, and so we can see where translation is actually happening on a very fine nanometer to micron scale. So we couple this with imaging, and this is some collaborative work with Keaton Holt and Elena Koslover at UC San Diego. So this is a human fibroblast. This is where the nucleus is, and this is the mitochondrial network. So we can use deep learning algorithms to segment individual mitochondria. We can follow them over time, and then we can see how they're actually sharing the information that they're processing with each other and changing their behaviors. So in the context of that marker that I told you about for translation, we can see that if we introduce a mutation into DRP1 that we know causes a neurodegenerative disease in humans, that it changes the mitochondrial network structure and it changes where and when translation happens on the inside, and that feeds back to tune the local ATP production of the network. So the big picture here for us is that information processing through the combination of regulation of the genome and the membrane dynamics is tuning the function of the mitochondria to cellular needs. We can also see this using cryo-electron tomography. This is some collaborative work with Kevin Rose and Jim Hurley at UC Berkeley. And we can use drugs like mitochondrial poisons to damage mitochondria, similar to how they would be damaged during aging, and then look very finely at what happens to the membranes and to the proteins. So here's the same set of images. And what we did is we picked many molecules of this protein ATP synthase that I mentioned that's so critical for energy production. And we could see that in the control cells, there were many proteins that were associated with the cristae, and we could assume that these are all producing ATP. But in the treated cells, most of the proteins had moved towards the edge of the mitochondria where they're not active. And so this suggests that we might be able to remodel mitochondria in a way that could kickstart ATP production and rescue the function of the organelle in older cells and in older organisms. So we've done a lot of work to establish the different pathways that regulate mitochondrial gene expression and dynamics, and I'm not gonna belabor this too much, but the big picture question that we want to move forward with is, can we target these pathways to reset mitochondrial network states in disease? So if we know what a healthy network state looks like and what a diseased network state looks like, or an older person's network state looks like, maybe we could reset it, and it would be like sending the mitochondria back to factory settings. And then the metabolism, in theory, would be more like a young person's again, or a healthy person's again. So one of the things we're doing is chemical library screening to identify compounds that can manipulate these pathways that we discovered. Okay, with that, I'll really thank you for your attention. I know I went from big picture all the way down to molecular level and back again, and this is the core group in my lab. We have many undergrads as well who are not in this picture, but thanks for your attention, and I'll thank my funders.